Hello everyone, I hope you're doing well. Today we're going to be discussing Nectin 1 and 4's role in Zlotogora-Ogur Syndrome. So let's get started. So what are nectins? The nectins have recently emerged as an immunoglobulin-like family that participates in calcium-independent cell adhesion. So the nectin family comprises of 4 members, nectin 1, 2, 3, and 4, which are encoded by PVRL1, 2, 3, and 4 genes respectively. And PVRL just means poliovirus receptor-like. They function as components of the epithelial junctional complexes, receptors for virulent entry, tumor suppressors, and molecules involved in immune reactions. Nectins share the same overall structural topology with an extracellular region containing 3 immunoglobulin modules, a transmembrane region, and a cytoplasmic region. Via their extracellular regions, nectins can actually homophilically and heterophilically interact with each other. And so they participate in the formation of cell-to-cell junctions cooperatively with or independently of another family of these cell adhesion molecules that are called cadherins. For our purposes, we're going to relate this to dentistry. So according to Yoshida et al., they state that nectins are found in the context between amyloblasts and stratum intermedium in the developing tooth. Amyloblasts are cells that are present only during tooth development that deposit tooth enamel, which is the hard outermost layer of the tooth forming the structure of the crown. Stratum intermedium cells show a high activity of alkaline phosphatase and act as a single functional unit in the enamel formation. So in the picture here you can see amyloblasts, and then down here you see stratum intermedium cells. So nectin 1 and nectin 4 are involved in Zlotogora-Ogur Syndrome, but how? So you might be wondering, first of all, well, what is Zlotogora-Ogur Syndrome? And judging by the pictures here, you can tell that it is cleft palate, which you've probably maybe seen or heard of already before, but this is just the name for the syndrome. And so we're just going to be talking about nectin 1 and 4 specific roles in this. And so first we're going to talk about how they act in a normal situation, and then we're going to talk about how they act when there's a mutation. So nectin 1's role, PVRL1 encodes nectin 1, which is a calcium-independent cell-to-cell adhesion molecule that plays essential roles in the essential step in the formation of adherence junctions and tight junctions. Nectin 1 is expressed in keratinocytes, neurons, and the developing face and palate, and various other organs. In mice that lack the PVRL1 gene, we have this expression of something called loricrin, which is this differentiation marker that is a major component of the cornified cell envelopes in the epidermis. What we end up seeing is a downregulation of it, and the newborn pups end up having shiny and slightly reddish skin. Cell-to-cell adhesion mediated by nectin 1 has also demonstrated to have a pivotal role in the nervous system. And so these observations are going to suggest to us that we are seeing pathological roles of PVRL1 mutations that could possibly occur in Zlotogora-Ogur syndrome. This syndrome results from homozygous loss of function mutations in the poliovirus receptor-like 1 gene. Nectin 1 contains an extracellular region with 3 immunoglobulin-like loops, 1 V type and 2 C2 types, a single transmembrane region, and a cytoplasmic tail. Patients who have PVRL1 mutations reported to date showed in each case that they were homozygous and that their parents were heterozygous for the mutant alleles. All 4 mutations identified in the syndrome cause truncated proteins, and they lack the transmembrane domain and the intracellular domain. And so a loss of those intracellular domain may disrupt the protein, and so we are going to end up seeing protein interactions that are necessary to initiate the cell-to-cell adhesion process are actually mutated. Nectin 4's role. Reflecting the fact that nectin 1 actually physically interacts with nectin 4, when we actually see a loss of function in mutations in the PVRL4 gene encoding nectin 4, we see the ectodermal dysplasia syndactyl syndrome 1. And this is synonymous with the Zlotogora-Ogur syndrome name. And so this is characterized by a combination of hair and tooth abnormalities like alopecia and cutaneous syndactyly, namely the phenotypes that largely overlap with those of Zlotogora-Ogur syndrome. And I'll be talking more about those phenotypes a little bit later on. So nectin's role in the Zlotogora syndrome, we'll talk a little bit more about that. So Zlotogora gave a review of this syndrome of syndactyly ectodermal dysplasia and cleft lip palate, which he proposed the disorder be called Zlotogora-Ogur syndrome. And so mutations in the human PVRL1 gene, which is basically the principal receptor used by alpha herpesviruses to mediate entry into human cells, are going to be implicated in cleft lip or palate ectodermal dysplasia syndromes, which include Zlotogora-Ogur Syndrome and something that's very related called Margarita Island ectodermal dysplasia. And so both of these are autosomal recessive disorders, and they're clinically characterized by unusual facial appearance, dental anomalies, hypotrichosis, and hypotrichosis, just little to no hair, palmoplantar hyperkeratosis, and that is just when you have hardening of the skin, thickening of the skin on like your palm. And then onychodysplasia, which is variable distortion of like the nail on the index finger. Syndactyly, which is 2 or more digits that are fused together. And then we obviously have like the cleft lip or the palate, which that just signifies like a groove or gap in the upper lip, right? And then in other cases, you see mental retardation. And so although the mechanism underlying that physiopathology of the Zlotogora-Ogur syndrome is still pretty much unknown, it's really been proposed that Nectin-1 is the cell-to-cell adhesion molecule that is preferentially expressed in the correct tanocytes, and that the mutations in that PVRL1 gene are going to abrogate NAP, which is nectin, affidin, and ponsin-dependent cell-to-cell adhesions. So those are very important in the formation of that cell-to-cell adhesion. So we're going to talk about Nectin-1's mutation. So mutations are located on the tryptophan 185 in the second immunoglobulin-like domain and the glycine 323 in the third immunoglobulin-like domain in the Nectin-1 gene. This ends up resulting in a nonsense mutation or a frame shift. Nectin-1 deficient mice do not develop cleft or lip palate, so the role of Nectin-1 in the formation of cleft lip and palate is still pretty elusive. But on the other hand, when we see TGF beta 3 deficient mice, and by the way, TGF beta 3, that just means transforming beta factor 3. And so anyways, when that cleft palate is going to be observed, and then the fusion of those palatal shelves is usually rescued by adding that TGF beta 3 complex. And so that ends up having the palatal shelves deformed again, right? And Nectin-1 is actually expressed in the medial edge of the epithelium. However, this expression pattern is lost when the TGFB3 deficient mice. So this suggests that Nectin-1 is actually involved in the initial adhesion of the opposing medial edge epithelium. So Nectin-1 is not only essential, but it's actually required for the fusion of the palatal shelves. So that's its role. When we talk about Nectin-4's mutation, so we see mutations in human Nectin-4 also cause ectodermal dysplasia syndactyly syndrome 1, and that is characterized by a combination of hair and tooth abnormalities like alopecia and cutaneous syndactyly. So mutations are located at the threonine 185 methionine in the second immunoglobulin-like domain and arginine 284 glutamine and proline 304 in the third immunoglobulin-like domain in the Nectin-4 gene. And so the arginine 284 glutamine mutation results in abnormal splicing, and the proline 304 mutation results in a frame shift. Other mutations like proline 212 arginine and valine 242 methionine in the second immunoglobulin-like domain are also reported. Some more molecular genetics of Zlotogora-Ogur syndrome. So in patients with the Margarita type of the ectodermal dysplasia, they actually identified a homozygous nonsense mutation in the PVRL1 gene. And so the author suggested the high frequency of this disorder on the Margarita Island actually has resulted from resistance of the heterozygotes to infection by the alpha herpesvirus. And so 2 families from Israel and Brazil described as having Zlotogora-Ogur syndrome, they also found homozygous mutations in the PVRL1 gene. So that was pretty interesting. And then for the orofacial cleft 7, Soren et al. Demonstrated a highly significant association between heterozygosity for the W185X mutation in the PVRL1 gene and sporadic non-syndromic cleft lip with or without cleft palate in northern Venezuela. So that was something else that I wanted to include. That was pretty interesting. And so we're going to look at this study really quick. And so we're going to talk about some of the clinical correlates that are associated with PVRL1 and PVRL4. So let's get on to that. So what we end up seeing here is some clinical features. So if you kind of look at this picture up here, you're going to see some sparse eyebrows and some kinky hair. You also see this concave nasal bridge with a low set of protruding ears, right? And then there is malar malar hypoplasia. And malar hypoplasia is basically just like the underdevelopment of these cheekbones right here that you can kind of see. We also see micrognathia, which is just an underdeveloped jaw. So you can see that more actually better in this picture. And then you also see scars from repair of the cleft lip palate. So you can see it right here and right here. We also see hypodontia, and hypodontia is just the absence of one or more teeth. So you can see he's missing some teeth there. And then you also see scars from the repair of the cutaneous syndactyl on the second and third and fourth fingers. So if you kind of look at his fingers, basically like the syndactyly was just when the fingers were fused, and so we had to get surgery to get them to all be free. And then you also can see tinea ungueum, and tinea ungueum is just like a fungal infection, so you can see that on his toes down here. And another thing, when they used a scanning electron microscope to look at his hair, they saw like pili torti, and pili torti is just fragile hair. And they also saw pili trianguli and a canaluculi, which is just unbrushable hair syndrome. So it's really hard to brush his hair. Now we're going to talk about the identification of the mutation. So basically this novel homozygous nonsense mutation in the exon 2 was found. And so let's look at A up here. And so when you look at A, basically what you're seeing is a pedigree of the family, and you see the identification of a novel nonsense mutation in the PVRL1 gene, which is right here. And so the position of this mutation is indicated by those arrows. And as we move down here to position B, we see the screening assay for the mutation as well with the restriction enzyme ZOL. So when they did PCR, they took some product from the wild type allele, which was 188 base pairs in size, and it was digested into 158 and 30 base pair fragments. While those from the mutant allele were just basically undigested. And so those 30 base pair fragments are not shown either. And now let's look at C down here. What we're seeing down here is the molecular structure of Nectin-1, and so Nectin-1 contains these 3 immunoglobulin-like loops in the extracellular region with a single transmembrane segment, and then it also has like the cytoplasmic tail. And so the numbers denote the amino acids located at the boundaries of each domain. And so 3 pathogenic mutations have been reported in the left cleft lip palate ectodermal dysplasia syndrome that are shown in the black, and so the new mutation is indicated down here at the red. Genetic tests. So in this very study that we were looking at, they actually looked at all the coding exons of the PVRL1 and some TP63 genes, which is involved in like tumor suppression. But we're not going to go too much into that. But including exon and intron boundaries, which were amplified by the PCR using gene-specific primers, and so these were primer sequences that were available upon requests. Another test that you could do, so it's like if you were a doctor or a dentist and you would tell your patient to go either see someone for these genetic tests or you would run them yourselves, you could do genetic testing of expecting parents. So if someone's pregnant, you could get like a prenatal diagnosis, and it can help to understand some of the risks better during pregnancy, right? You can also use a karyotype, so you can compare like 500,000 snippets of human DNA that scientists from the University of Bonn put, and what they ended up finding was that they had a genetic variant on chromosome 8 that occurs with significantly higher frequency in people with cleft lip and palate than in a control group, so you can get tested against those karyotypes. Another thing you can do is use fluorescence activated cell sorting analysis, which is a special type of flow cytometry, and it basically provides a method for sorting a heterogeneous mixture of biological cells into 2 or more containers, one cell at a time, based upon specific light scattering and fluorescent characteristics of each cell. So you can kind of look at each cell under the light. And you can do whole genome homozygosity mapping, which is a strategy that is common used to locate identical chromosome regions in homologous chromosomes that are shared in patients in families that are affected by autosomal recessive disease. Then we're going to talk about treatment or care. And so management of clinical manifestations associated with ectodermal dysplasia syndrome presents a unique challenge. And so early diagnosis, sympathetic and a rational attitude, and a multidisciplinary treatment are very important. And so the patient should be referred to like medical practitioners for a systemic examination, so ophthalmologic exams, audiometry, speech therapy. They should also be examining like the genitourinary tract, the cardiovascular system, the GI, the respiratory should all be carried out, and even doing like an IQ test. And so the dentist's role in this could be possibly to do like a surgical closure of the cleft lip, and it's usually indicated at 10 weeks of age, and so an oral surgeon could possibly do this. And the closure of the palate is accomplished by 12 months and 2 years of age. And so the primary purpose of completing the palate closure by 2 years is really to facilitate the acquisition of normal speech, because this is going to correlate with the age at when children really begin to talk. And so you can also do orthodontic treatment that's interceptive and plan at late primary and mixed dentition period to counter developing anterior or posterior crossbite and traumatic occlusions. And so these are my references. If you guys end up having any questions about this or want to know more, please feel free to email me. That's it. I hope you guys have a wonderful day. Take care. Bye.